We'll now study some of the organelles found inside of the cells, and these can be divided into two categories. The first category we'll look at are membrane-bound organelles, or membranous organelles. And here's a list of those and where you can find them on a typical cell diagram. I won't go through this screen, but we'll just tackle these one at a time. Starting with the nucleus, sometimes called the cell CEO, the Chief Executive Officer, because it does control the cellular activities and it stores the genetic information. And you may be used to thinking about and talking about chromosomes, but really those only form during periods of cell division. Most of the time, inside the nucleus, the DNA is just a tangled mass of a very, very thin strand of genetic material called chromatin. And because we won't be studying mitosis, prophase, metaphase, anaphase, and those different stages of cell division, we won't really see any chromosomes in this course. Most of our cells are out doing their jobs, not dividing, just delivering oxygen, creating mucus, etc. The nuclei also have a nuclear envelope. It's just like a plasma membrane around the nucleus. It has pores in it so things can enter and exit the nucleus. And a nucleolus, a small dark staining body within the nucleus. And this is where ribosomes are made. And ribosomes, we'll talk about soon, make proteins, which do almost everything for your body. Nucleolus is worth knowing and understanding because if you were to look at something like this, and I told you I wanted you to show me a cell, well, there's all these round shapes in there, and you might not know where to start. But here's the tip. A nucleolus is the smallest thing that you're going to be able to see with a light microscope. If you get some new fancy technology, a scanning electron microscope perhaps, you can see much smaller things. But for our purposes, look for the smallest dot. It's probably the nucleolus. So in this case, here is a nucleolus, which makes this the nucleus, which makes this the cell. Everything else that you see there is, in this case, a follicle inside the ovary that is helping that egg cell grow. So you can do the same thing off on the other side of this image. If you find the smallest possible dot, you can find the nucleolus. That will help you know where the nucleus is and where the cell is. So just something to keep in mind. Mitochondria are the powerhouses of the cells. They have these inner folds called cristae, very classic mitochondria appearance. And the spaces in between those is the matrix. And that's where sugar molecules and oxygen are being turned into ATP and CO2. If you want to know why you're breathing right now, this is a big reason for that. You're bringing that oxygen in to help make ATP, but as a byproduct, CO2 is also created, and then you need to breathe that out. ATP we won't study in much detail, but it's the cellular unit of energy, which means like glucose, these sugar molecules are important, but the cell can't really do anything with them. It's like if my kid wants to roll out the Play-Doh, he needs a little small roll tube. If I give him a steamroller, it's kind of the right idea, but it's not going to do the job. A glucose molecule is this big structure that isn't going to do the job. It needs to be converted into a tiny little ATP. That's really all you need to know about ATP in this course. Mitochondria also contain their own DNA. Maybe you've heard of mitochondrial DNA is good in researching evolutionary relationships and things like that. Endoplasmic reticulum, kind of a long name here, workshop, storage area, shipping depot of the cell. So we'll revisit those. Let's just look at what it looks like. It's this network of membranes that forms fluid-filled chambers. And substances can cruise around inside those chambers to be transported within the cell. But the endoplasmic reticulum is also busy synthesizing stuff. So the rough ER is studded with ribosomes. And we mentioned already that they make proteins. So the rough ER makes these proteins. In the smooth ER, well, it's not making proteins since it has no ribosomes. So it does a lot of stuff, but including making lipids. We won't learn a ton about lipids, but we've already learned about phospholipids. So if you want to know who's in charge of making new phospholipids to replace the old ones that need to be taken out, smooth ER. Cholesterol is a type of lipid. So if you want to know who's making sure the cell membrane stays the perfect amount of fluidity, smooth ER. And steroids, other steroids besides cholesterol would be like steroid hormones, like testosterone and estrogen. The testes and ovaries have a lot of smooth ER in their cells to make those steroid hormones. So smooth ER, pretty good, making all different kinds of lipids. The Golgi apparatus is the official post office of the cell because although the endoplasmic reticulum, which is seen in the bottom of this image, can move some things around, if you really want to send something a long distance, you need to use the Golgi apparatus. And what it's doing is packaging products for transport. So like the endoplasmic reticulum sends a lot of substances to the Golgi apparatus because it's doing the final sorting and packaging. And the Golgi apparatus will package stuff into these little spheres called vesicles. And then there's three possible outcomes here. One option is the vesicle becomes a lysosome, and it's going to stay inside the cell. We'll talk about lysosomes on the next slide. A second option is that the vesicle fuses with the cell membrane. So it's basically sharing its phospholipids with the cell membrane, as we said, to replace those that are damaged or perhaps have been used through endocytosis. When the cell pulled something inside, it captured it with its cell membrane, and then that cell membrane piece goes away. And a third and final option is the vesicle could become a secretory vesicle. So it will fuse with the cell membrane, but in this case, the goal here is to release its contents through exocytosis. Maybe there's a bunch of hormones in there, for example, that are sent out into the bloodstream to signal other cells. Let's look at those lysosomes again. They're these membranous sacs, and they're full of digestive enzymes. So they can break down old or damaged organelles that need to be replaced. Lyse means to cut. L-Y-S, to cut. So that's a good one to know. It'll come up a lot in the course. So you can cut up the old stuff, the damaged stuff. Bacteria or viruses or foreign materials can get cut up through lysosomes. In a previous video, we saw an amoeba through phagocytosis eat some paramecia. And the paramecia didn't seem to notice until suddenly they did. That's when the lysosomes started to digest the paramecia. The other thing lysosomes can do for humans is autolysis, which is called cell suicide or programmed cell death. And that occurs like when, as embryos, we have our little paddle hands and our tails and all these extra features that we want to go away so that our paddle hands become fingers. So autolysis will kill the cell. Many cells will be killed to create our fingers and get rid of our tails and stuff. Peroxisomes are created a different way than lysosomes, but they're kind of similar. They're membranous sacs. In this case, they have some oxidase enzymes. So these are going to take care of harmful substances like alcohol, formaldehyde, and free radicals, these very reactive chemicals. So they're just trying to keep the body safe from dangerous chemicals. And like our bodies make hydrogen peroxide, for example, as a waste product. So peroxisomes will take care of that. If you have a cut and you pour hydrogen peroxide on it, it'll foam and bubble. And that's peroxisomes in action. They see the hydrogen peroxide, their job is to get rid of it. And they do that by cutting it down into oxygen and water. So that's why you get that foaminess. Your peroxisomes are trying to convert that into something safe for your body. That's their job. Okay, now let's do these non-membrane bound organelles. Here's a list for you to review later. Ribosomes, we already said, create protein. So they're these little factories of the cell creating proteins. They can be free, floating about in the cytoplasm, or fixed, meaning they're embedded in the rough endoplasmic reticulum. Then we have the cytoskeleton, which you could think of perhaps as the city planner of the cell, because it creates this internal framework that kind of determines where things are and gives it support. It also allows things to move about within the cell. And the cytoskeleton is made of 3 structures: microfilaments, intermediate filaments, and microtubules. So those are in order from smallest to largest. Microfilaments are especially found just under the plasma membrane, so they're really helping your cells keep its shape and have the right amount of movement to them. So they brace it and strengthen the cell surface. Think about especially like erythrocytes, your red blood cells banging around inside your blood vessels. They need quite a bit of support. Intermediate filaments are made of proteins with high tensile strength. Tensile strength just means if you put tension on them, they can handle that. So if you're pulling or stretching them, they can handle that movement. Microtubules are hollow tubes made of protein, and they will anchor the organelles and allow the organelles to move about within the cell. And they're also very important during cell division. They create these mitotic spindles, which will help separate all the different components of the cell during division. The cytoskeleton, though, is not a static structure. So hopefully my video here will play. Yeah. So the orange and yellow lines there are all parts of the cytoskeleton. And if you pick just one area of the cell, you can watch and you'll see new pieces emerge, old pieces go away, and there's especially a lot of activity on the periphery just under the plasma membrane. So, just to remind us, just like our real skeleton, which we'll talk a great deal about later, how our skeleton is living, it's changing, so too is the cytoskeleton. Okay, centrosomes and centrioles. The centrosome means central body, because remember we learned that soma means body. This is somewhere near the center of the cell, pretty much a glob of protein. And remember, microtubules are made of protein. So this is where they're made. Inside this glob are 2 centrioles. Those are bundles of little short microtubules that make these cylinder shapes. And those are important for organizing the microtubules in your cells and creating that mitotic spindle to help pull things apart during cell division. Okay, so we're about to cover this important concept. So we need a little background information on surface area to volume ratios. Let's not get too complicated, but let's acknowledge that if you take a desert jackrabbit and a desert fox and someone whose ancestors are from Sudan in this case, and you compare them to an Arctic rabbit, an Arctic fox, and an Inuit person, what you'll see is that the appendages are different lengths. Arms and legs, or ears and limbs, are going to be different sizes, and that's really trying to assist with thermoregulation. We actually do this behaviorally. Like if you're very hot, you kind of increase your surface area by sprawling out. And if you're very cold, you decrease your surface area by assuming a fetal position. So the body plan of these different animals and humans can do that based on the surface area to volume ratio. In other words, perhaps these two people are the same weight or have the same internal volume, but they're very different in their shape. So too is this cube, the same volume as this wall. Both are made of these 8 units of matter, but their surface areas are very different. Okay, so why do we care about this? Well, one thing is that cells can't grow to enormous sizes, because if you had a big blocky cell like this, the very center of the cell is like where the nucleus is, for example, and you need to make sure that you can get nutrients and collect waste from every part of the cell. So an enormous cell will have a surface area to volume ratio that will not be conducive to its survival. But more apt to what we're about to talk about with microvilli is that the other thing surface area can do when you have a lot of it is help with absorption. Okay, let me say something like, if you had a spill of water on your tabletop and you took a huge sponge that was, I don't know, a big ball shaped like this, and you put it on the spill, only the bottom of the ball would touch the spill, right? That's not very much surface area to help absorb the spill. But if you took that same ball and you squashed it into a big flat disc shape and you drop that on the spill, oh yeah, super good absorption. That's what we're talking about here when we talk about microvilli. If cells had just flat tops, and many of them do because they don't need to absorb, that wouldn't be very good at absorption. But cells that need to be involved in absorption, for example, these cells inside your intestines have these things called microvilli, all these little finger-like extensions of the cell membrane, because those are going to help nutrients get absorbed into the cells. They don't move around, they just sit there, but they allow more absorption to take place. Just like a sponge that's designed properly for your spill will be more efficient at absorption. Not to be confused with cilia, because cilia do move. These are whip-like extensions on the free surface of a cell. So as you can see here, they're all kind of at the top, the apical surface of the cell. And they're very different. So these aren't extensions of the cell membrane. These are made from microtubules. And in humans, the goal of cilia is to move stuff along the surface of the cell. Like when you suddenly feel in the back of your throat a little bit of some mucousy, sludgy slime in the back of your throat, how did it get there? Well, you've been breathing in and out all day, and there's a bunch of mucus that's been trapping crud from the air. And these cilia have slowly been whipping or beating the mucus up until it reaches the back of your throat. Now you can spit it out, or you can swallow it, and your stomach acid will kill anything nasty in there. So the cilia are always beating to move things in one direction. They're not cilia that let your cells swim around in your body. They're moving some substance over the surface of the cell. Another example would be in the fallopian tubes. If you want the egg to travel down the fallopian tube towards the uterus, you need to make a current to push it the right direction. And cilia do that, moving a fluid that carries the egg down towards the sperm. Not to be confused with flagella. Also whip-like extensions, but in this case, we're just saying that they're on sperm, and now you really are trying to move the sperm cell. Here's a single sperm with its flagella. Here's inside the testes where you can see all these sperm and all the little flagella that are being formed. Okay, last thing, we're going to take a peek at this video so we can see a few of the structures we talked about in action. Here we can see the phospholipid bilayer, lots of movement in the phospholipids as they kind of shuffle about. Things moving about on the surface of the bilayer. And we can see some cholesterol molecules embedded in the cell membrane to help keep it perfectly fluid, not too goopy, not too rigid. Lots going on with different proteins and other structures completing their functions on the surface of the cell. This is kind of the space in between our cell and another cell. Now we go into our cell. More action happening inside. There's some proteins. And we can jump ahead a bit and see some of the cytoskeleton. Here are some microfilaments. Remember, a lot of those just under the surface of the cell to give it some strength. But we had mentioned already that the cytoskeleton, like our own skeleton, is very dynamic, meaning it's constantly changing. So we'll see these microfilaments being assembled and being broken down. In addition to microfilaments, we had intermediate filaments, and then the largest are these microtubules. So you can see they're hollow cylinders, much larger than the microfilaments, and again being built up and being broken down. One of the functions that microtubules have is the ability to assist in the movement of organelles within the cell. There's what I call a blueberry mosquito, but is really a vesicle traveling down a microtubule. So, is it a lysosome? Is it headed to the plasma membrane to help replenish the phospholipids there? Maybe it's full of hormones it will secrete. Who knows what that vesicle is up to? There's a mitochondrion, singular, cruising down the microtubules. And near the center of the cell is the centrosome, that glob of proteins. Embedded within are the centrioles, helping organize all these microtubules. And some pores in the nuclear envelope. We can also take a look at the Golgi apparatus, seeing how it's receiving some vesicles from the endoplasmic reticulum. It's going to process, repackage, sort those products, and then secrete these vesicles. There goes one now. I think the blueberry mosquito is a good place to finish.